So yeah, as Katie said, I was going to do this talk in two parts today. So these are sort of what I'm hoping to cover in the two parts. The first part being an overview of TEM, a description of normal ciliary ultrastructure, the class 1 defects that allow you to confirm a PCD diagnosis, and then where EM fits into the current American and European diagnostic algorithms. And then in part 2, maybe some of the more complicated and challenging parts of EM and some of the future directions for research. So first of all, what is transmission electron microscopy? Essentially, it's like a light microscope, only instead of a light, you have an electron beam. The whole thing is in a vacuum, and you project through a specimen, which causes essentially a shadow onto a plate so that you can see what you can see inside your specimen that you put into the microscope. Your average clinical electron microscope looks something like this. I'll just turn my laser pointer on. Looks something like this, and this is one at the Brompton. But you also nowadays get much bigger, fancier electron microscopes, which are as big as a room. And this is an example here with a person standing by it. So there's a huge spectrum of different electron microscopes, and depending on the microscope and the technique you use, allows quite good resolution of structures, and much better resolution of structures than you can get through using standard light microscopy. So this is the sort of thing you can see at different sizes. So a eukaryotic cell, and here is a cell here about 100 microns, bacteria here at about 10 microns, quite topical viruses at about 100 nanometers. And I thought it was nice to put this picture in. So this is from our labs at the Brompton of SARS-CoV-2 taken by Andrea Pinto, binding to microtubules and binding to cilia in the airways, so you can comfortably see a virus. And important for us, you can see the cilia and you can see the internal structure of the cilia. So this is what an epithelial strip looks like under an electron microscope. You have individual cells here, you have a nucleus, and pointing out the top here, you have the cilia projecting from a basal body, and that's a bacterium there. And if you look at those in cross section, then you get this sort of appearance where you can see the classic 9+2 microtubular organization of the cilia. And if you zoom in further, then you can see these dynein motor proteins which cause the cilia to beat and move. So this is just an overlay of the important structures that we're going to need for this talk to understand ciliary ultrastructure. You have 9 outer microtubular doublets, a central pair of microtubules, these outer dynein arms, which are the powerhouses that cause the beating of the cilia, the inner dynein arms, which are a little bit trickier to see because they repeat less often as you move down through the cilium, but coordinate also the power of the beating, but the pattern of the beating. The radial spokes, which are like the wheels or the spokes on a wheel, and hold the structure together and communicate messages between the central pair and those dynein arms. And then these bits, which is the nexine dynein regulatory complex, which does exactly what it says and regulates the dynein, and therefore regulates the way in which the cilia beat. The first time that electron microscopy was used to diagnose primary ciliary dyskinesia essentially was in the first description of what was then called a motile cilia syndrome. And it wasn't actually in airway cells, but was in sperm tails. And it was by this guy here called Bjorn Afzelius. And there's a great review article here if anyone is interested in the history of PCD discovery and diagnosis with pictures of all of these famous people with their microscopes. But essentially what he described is that in the sperm tail, this is a normal sperm tail with a structure that is very similar and very well preserved compared to the airway epithelial cells. You have the 9+2, you have the outer arms and the inner arms. And in this patient who had bronchiectasis and also had sinusitis and had infertility in a motile sperm, they had none of the dyning arms. So it goes all the way back to 1976, us being able to see these structures and use them in the diagnosis of PCD. And of course, this is confirmed in airway cells. You see the same thing in airway cells, the same thing was seen in that paper. And this is an example here. So you have the structure of the 9+2 and complete absence of the outer and inner dining arms. And because those outer and inner dining arms are missing, and those are the power of the cilia moving, then you tend to see with that complete lack of movement or very little movement by high-speed video microscopy. And we now know that these combined outer and inner dining arm defects are mainly caused by gene defects in things that are related to dyning assembly within the cell. So this is a list of all of the genes we know that cause this kind of defect. To date. So what we're seeing, and I think this is important for later, what we're seeing with the absence of outer and inner dining arms is the effect of these assembly proteins being absent. And I thought I'd just include for all of these defects a few more examples. So this is another example taken from another lab. So quite often things look slightly different depending on how the lab has processed the samples or the staining used, the timings, or how it's been sampled. But you see a similar kind of thing, that you get the 9+2, and I think I've got a zoom in here, and really nothing where the dining arm should be, and that's fairly consistent on most of the cross sections. Occasionally you see a little thing which might be an arm, and that's not completely unusual, but in the majority of the cross sections, on the majority of the microtubular doublets, the inner and outer dining arm are missing. And here's another example. So these cross sections you can't see quite so well through the microtubules, but you can still see the absence of the outer and inner dining arms in this other patient. So what are the, so this is the inner and outer dining arm defect, and that occurs in about 25% of people who are diagnosed with PCD by transmission electron microscopy. And this table is the characteristics of ultrastructural defects from 9 studies, which are in the European Respiratory Society guidelines for diagnosis of PCD. So in making these guidelines, we looked at, we did a literature search for all of the studies that had done a consecutive diagnostic protocol for people with symptoms of PCD, and then included studies where a diagnostic conclusion had been made, and that TEM was one of the outcomes. And we found 11 studies in the literature that met the strict inclusion criteria for the European Respiratory Society guidelines. And out of those, 6 or 9 of those studies had included what the ultrastructural defects were. So that's what this table is essentially, is telling us what the most common electron microscopy defects are in some of the larger cohorts of patients with PCD that have been reported to date. So the most popular or the most common is the outer dining arm defect, and so I'll go on to talk about that one next. So the outer dining arm defect, very similar to the outer and inner dining arm defect, only the inner dining arms are present. And I've really zoomed in here on one of the microtubular defects. You can see that the inner dining arm is quite clear there, and there's really nothing here where the outer dining arm should be. And again, there's a whole host of genes that we now know cause these outer dining arm defects, and we know which parts of the structure of the outer dining arm those code for. And interestingly, there's a little bit of, you can tell sometimes whether what you're going to identify in your genetics is going to be heavy chains or docking complexes. So this is a map of the outer dining arm, and these are the heavy chains that are on the end, and this is the docking complex that attaches the arm to the tubule. And this is a patient with a docking complex defect. So you see really nothing at all there because the arm hasn't been able to dock at all. DNH5, which is the most common cause of PCD and the most common ultrastructural Itodine Arm Defect, you see that docking complex is still present. So you get a little stubby bit which is still present, but you don't have a full Itodine arm, and that still constitutes an Itodine Arm defect. And here's another example of a DNH5 defect. So you can see you get little stubby bits, but if you compare the Itodine arm, or what should be the Itodine arm, to the Inodine arm, it's not as full. You've got a lot of space, and that happens on the majority of the arms in the majority of the cross sections, defining it as an Itodine arm absence. So for each of the defects I've described, I've showed a couple of pictures in this talk, but we do have a library of pictures of EM defects for people wanting to compare their own samples, or just for interest, and that's on the BEAT-PCD website. So if you go to the BEAT-PCD website under resources and click on example images of defects, all of the common and some of the rarer PCD defects, there's a library of those that you can look at your leisure if you wish. So the final hallmark defect for PCD is this inodine arm with microtubular disorganization. And in this defect, the inodine arm is missing, so the Itodine arm in general is intact, but the more striking feature is you have this whole collapse of the structure. So instead of the nice 9+2, the structure is fairly collapsed. You sometimes get a double central pair. This is from a paper describing CCDC39 and CCDC40 defects, which is the main gene which causes this. And interestingly, this figure here is from this lady's fallopian tube. So you actually see the same ultrastructural defects by EM in the fallopian tubes as you do in the airways. And here's another example here. This video isn't working, but essentially you see some movement of the cilia by high-speed video microscopy. By immunofluorescence, you see an absence of gas8, which is a marker for the nexin-dynein regulatory complex, and you also see an absence of DNA Li1, which is a marker for the inodine arm. These defects is becoming increasingly clear from a number of studies are associated with a worse outcome. So although there's still a little bit of ciliary movement preserved, and those Itodine arms, which are the main powerhouses of movement, are still intact, people tend to do worse and have a worse progression of their lung function over time when diagnosed with these defects. So these defects caused for a while a few problems amongst the PCD community, because it's a rare disease and people have really developed their own way of making the diagnosis by electron microscopy within their own labs, in their basements with their electron microscopes. And it came to a point where everyone was calling this something very different. So you can see, when you see this microtubular disorganization and lack of inodine arms, you have spaces between the doublets where there's no nexin-dynein regulatory complex, and therefore people were calling this nexin-link defects. The inodine arms are missing, and there was some concern that the microtubular disorganization was secondary to infection or inflammation, and therefore they've been cited many times as inodine arm defects. The radial spokes, as indicated by this yellow thing here, are missing, and you can imagine how they are missing because the whole structure is collapsed in on itself. So they're sometimes referred to as radial spoke defects. And then other labs find it difficult to see the inodine arms and to comment on their presence or absence, and therefore refer to these as microtubular disarrangement defects. So this was beginning to cause a bit of a problem when you go back through the literature and when you're talking to one another, because actually we're all talking about the EM from a patient with CCDC39 or 40, and we're all using very different terminology. And therefore, we started within BEAT-PCD to try and organize a consensus statement where we had 18 different labs from, I think, 14 different countries. So America, Canada, many countries in Europe, Belgium, Germany, Poland, Spain, Australia. So many of us got together who were doing PCD electron microscopy and wrote essentially a consensus statement on what constituted an EM defect and what we maybe should be calling them. So that was done in a few stages, the first of which being a expert consultation, and then we did an anonymized Delphi survey where we all voted on what the definitions would be. Where there was any disagreement, we went back to the literature and discussed that. We had quite a few meetings. We then consulted more widely within the PCD community, one of the BEAT-PCD meetings, and finally we exchanged a lot of photographs and grids from patients and checked that our definitions were working and that we could all agree on what the definition was, having written the guideline. And I don't think it's necessary to read this, but essentially these three defects that I've talked about so far, the outer arm absence, the inner and outer arm absence, and the Itodine Arm and Microtubular Disorganization defects, we refer to as the class 1 defects. And essentially they need to be seen in the majority of cross sections on the majority of microtubular doublets in order to be called. And we've put in a caveat here because there was some difficulty sometimes with seeing the Itodine arm, that if you can't tell if it's an outer or an Itodine arm defect, then it's okay to call it an Itodine arm defect plus or minus an Itodine arm defect because of the problems with seeing those structures. So we also tried to define the sort of quality of the sample that was required to make the diagnosis. And class 1 defects are, we say you need to be confirmed on the assessment of at least 50 axonemes within transverse section. And that helps to, I think more important than the number is that a number of cells are looked at and a number of different positions within the cilium, so at the base and at the tip, not just in one plane of view, but at least doing 50 axonemes means that there's a high chance that you're going to end up in all those places. And within those 50 axonemes, then cross sections should have a really good ciliary membrane and be able to clearly see the structural features. And I've just got an example here. So this is an example of something that you wouldn't count if you were doing diagnostic electron microscopy, because the membrane is completely fallen apart. It's a necrotic cell. It's probably not been processed particularly well, or it's been sitting around before it's been processed. And it's really tempting to count these because the membrane's not intact, the stain gets in really well, and therefore you can see the cilia really clearly. But if you see a defect in these cross sections, you really can't be sure whether that is a defect due to it being PCD, or if that's a defect due to the fact that the structure's been affected by the cell dying, essentially. And the other thing is that the cross section should be clear, and you should clearly be able to see the structural defects. So this is another picture here. So you might count this one and this one and this one, but maybe you wouldn't count this one because you can't really tell if the Itodine arms are there or not, because it's a bit blurry because of the angle at which it's sitting. But what you can do is look at the 9+2 microtubular arrangement within this cilium. So you can say, I think quite safely, that this is 9+2, but maybe not comment on the arms. And as a result, you probably will end up under the microscope counting a lot more cross sections for microtubular arrangement than you would for dyneins. So these three defects I've described are the class 1 defects. If you find one of these defects, then that is definitive, it's positive as a diagnosis for PCD. So that patient has PCD without the requirement for any further tests, in the same way as if you find a non-ambiguous biallelic mutation in a PCD gene. And that makes it a useful test for confirming the diagnosis of PCD. And within the European Respiratory Society guidelines, EM sits here essentially in the middle. So once you've screened using nasal nitric oxide and high-speed video and screened out people who are unlikely to have PCD, then electron microscopy can confirm the diagnosis. And if electron microscopy is normal, then you proceed to genetic testing. The American guidelines are slightly different in that genetic testing goes higher up the algorithm, but essentially electron microscopy is still key down in this arm. So again, you do nasal nitric oxide and screening on clinical history. And if you don't find a certain pathogenic gene mutation, or that needs to be confirmed because it's a variant of unknown significance, or it's a single variant, or no variants are found, then you go on to do electron microscopy and again recognize ciliary ultrastructure defect is diagnostic. So in both guidelines, a recognized ultrastructural defect is diagnostic of PCD. And this is the evidence for that, which was assembled in the European Respiratory Society guideline. And electron microscopy actually has the most evidence of any of the PCD tests when we went through the literature, and that's because it's the oldest. It's because it's been around for a very long time, and therefore there is a lot of evidence for it as a diagnostic test for PCD, because for a very long time it defined the disease. So looking at all of the studies that met our inclusion criteria for sensitivity and specificity of PCD, you can see that 92 to 100% specific for a diagnosis of PCD, which means you're really unlikely to get a false positive. So if you see an Itodine Arm Defect, it's very, very unlikely that person doesn't have PCD. So the presence of a Hallmark defect is diagnostic of PCD. It is, however, not that sensitive. So if you get so 70% to 100% of so 70% of patients will have normal ultrastructure, but it doesn't mean that PCD is excluded. So you can't exclude PCD using electron microscopy, and that's been known for a very long time. So it was first described, I think, in the early 1980s, that you can have PCD with normal ultrastructure. That can occur in up to 30% of cases, and I think we're learning about more and more cases that have normal ultrastructure as genetics is advancing. And the largest cause of that is the dynein heavy chain DNH11, which is the second most common PCD gene. So it's really, really important to always remember when you're doing electron microscopy that if you get a normal result, that doesn't exclude a diagnosis of PCD, and other tests need to be done, especially if there is a very strong clinical history for that patient. So that's the end of part one, the overview of the EM, the description of normal ciliary ultrastructure and the class 1 defects, and how EM fits into the diagnostic algorithms. In the second part, I'm going to go on to talk about the class 2 defects, where electron microscopy, instead of being a diagnostic test on its own, it needs to work in conjunction with all of the other tests, with the genetics, with the high-speed video, with the immunofluorescence, with the nasal nitric oxide, or whatever tests you have available at your centre. But I think EM within that context can be extremely helpful to help add evidence that it is that PCD is the cause of the patient's symptoms. So I think part 2 is possibly where EM gets more interesting for those of us that spend time at a microscope. But I thought I'd pause before going into that, because it's quite a long talk, and run a bit of a quiz. And I don't know if you want to write answers in the chat or if you just want to think to yourself, but I've got 3 images and I want to know what you think this defect is. And I actually can't see the chat, so I also can't turn off my laser pointer, which is really annoying me. Okay, I'm going to assume that some people have made a decision on what defect that is. So this is an inner dynein arm and microtubular disorganization defect. You can see from the collapse of the structure and the lack of inner-dining arms, although we obviously wouldn't diagnose it from this photograph alone because we need to look at 50 cross-sections. So this is number 2. If anyone wants to write in the chat what they think about image number 2.
I'm not sure if you can see the chat now, Amelia. Somebody said outer dining arm and inner dining arm.
Yeah, I'm getting lots of outer dining arms and inner dining arms coming in thick and fast. I agree. And then the last one, which you might be able to get by process of elimination. Just give a couple of minutes, maybe seconds. Yes, so this is an outer dining arm defect, and you can see the inner dining arms hopefully fairly prominently still present, but nothing here in these outer dining arm sections. Does anybody have any questions at this stage? I don't know if you'd like to break for questions before we go into the second half. I'm happy to take any questions.
I can't currently see any questions in the chat.
Okay, and I can carry on.
I think we'll carry on. Are you, in the worst case, are you happy to be interrupted? No, it's probably tricky.
I'm happy to be interrupted. Yeah, I don't.
I think we'll
can't see the chat, so you'll have to actually interrupt me. But yeah,
that's fine. We can do that.
I'm just struggling with. Okay, so this is part 2. So this is part 2, which is the class 2 defects, those which indicate a diagnosis of PCD in a patient with clinical symptoms of the condition, if they're consistent across more than one sample, within that sample, and consistent with other results from other diagnostic tests. So there's lots of caveats of this, but I think they're interesting and can help with diagnosis. So to be able to recognize these defects, and they're discussed often as being possibly unreliable or just normal ultrastructure, but to be able to recognize them, you really have to have maybe a slightly better understanding of the ultrastructure of cells and of cilia. So I'm just going to talk you through a couple more bits of normal ciliary ultrastructure before we move on. So an important thing to recognize is that the cilium, or the axoneme of the cilium, is not the same all the way through. So the bits we often look at are in the middle, and that's the majority of the axoneme, and they have this 9+2 structure. When you get to the very top, the cilia narrows and you get single tubules instead of the microtubular doublets, and you maintain the central pair, and eventually they disappear as well. At the very bottom, you have an absence of the central pair. When you're really close to the cell, you have these Y-shaped linkers, which sometimes orientates you that you're near the bottom, you're within the proximity of the cell, and that's why your central pair is missing. Your microtubular doublets don't have dining arms, and then as you move into the cell, you have centrioles, you have your you have these triplets. So instead of doublets, the microtubules form triplets. So that's essentially the ultrastructure as you move up through the cilium. And if you look in longitudinal section at cilia, you can see your microtubular doublets on the outside here. You can see the You can see the central pair in the middle. You sometimes, if you're really lucky, can see a little bit of the radial spokes. And then at the base, you see the basal foot, which has a foot which projects usually in the direction of the ciliary beat. And then out of the bottom, you have a rootlet which anchors the cilia, and that's striated. So you see striations there. You can, by looking at the cell and by looking at the cilia, you can usually get an idea of whereabouts you are. You can orientate yourself within the sample. And a really useful way of doing this is to use the microvilli. So the microvilli come sort of a third to half the way up through your cilium. And so if you're at the top, in general, you don't have any microvilli around. And if you're near the bottom of the cilium, then you have lots of microvilli around. And that's quite helpful for recognizing where you are. And hopefully I'll show you later on that some of the defects only occur in some sections of the cross sections. So if you look at lots and lots of cross sections from just within the region of the microvilli or from just near the top, near the tip, then you miss things and you report normal ultrastructure when it isn't. So this is a healthy sample, and I just thought it was helpful to again look at all these features that I've just described sort of within the context of a sample. So this is within the cell where you see the cartwheels, you see the triplets. This is a cross section just coming out of the cell where you see you don't have the central pair yet, and the microtubular doublets don't have an itodine arm. The same again there, and then as you move up, you start to see the cross sections as you would expect to see them. And I'm covering this because people relatively frequently send me photographs of things like this and ask if it's a defect. So I think it's useful to know that it's not, and that it's normal to see that. And then once you're outside of the area of the microvilli and you're up near the tip, again you see these singlets and you see a narrowed axoneme, and you know you're near the top of the ciliary axoneme. And I think it's useful to think about that and think about where you are within your sample, not just oh good, there's some cross sections. And here's another example. So you can see right at the base here, no central pair. These are a nice picture of those triplets there. And every so often you come across something like this. And this is a compound. So this is two axonemes which have fused together. And in you and I and normal samples, healthy samples, you see this sort of between 1%, maximum of 10% of cross sections, and you see a number of secondary defects like this, and that can be for a number of different reasons, but you definitely see ciliary ultrastructural defects in a proportion of samples as a result of bacterial viral infection, as a result of inflammation, sometimes as a result of sample processing, because the cells have died, because the cells are old, and because there's other processes going on. So not all ciliary defects are primary, and not every ciliary defect you see is primary ciliary dyskinesia. So this is an example of a compound. This is a really extreme case of compounds, and it's unusual for you to see this, but it's reportedly to be secondary to inflammation, that you see lots and lots of axonemes within one membrane. You also see things like this in unhealthy samples. So you'll see little gaps within the B tubule, which I always call Pac-Man cilia. Was reported once to be a cause of PCD, but has never been proven genetically or otherwise. You see some disarrangements. You see in an unhealthy cross-section an absence of the central pair sometimes. You see absence of some of the outer microtubular doublets. You see extratubules that show up within the membrane. And the important thing to bear in mind is whether this is consistent across the sample, or whether it's just in one area, and what the associated cell looks like. So is your associated cell unhealthy, or is it healthy? And this is an example here. So this is a relatively healthy epithelial strip, but within a sample you get lots and lots of cells like this that are dead. You can see they're vacuolated. They look like, I always say, ghost cells because they don't contain the same structures. And there's loads and loads of tempting cross sections here that you'd quite like to look at, but there really is no point because they can be like this. They can contain loads and loads of defects. So you can see that the membranes are no good, and you can see lots of absences of the central pair. There's a 1 pair missing here. There's a both of pair missing here, and that's just because that cell is dead. And that's just an example there of a processing artifact with really bad processing where really you can't see any structure whatsoever. So it's not always down to the sample that it's unhealthy. It can sometimes be down to the way it's processed. So now moving on to the class 2 defects. The first one I'm going to talk about is the central complex defect. And the central complex defect is associated on high-speed video with these circular motion, which I don't know if I can get it to play. Anyway, associated with the cilia circling rather than doing a back and forward motion. Patients don't tend to get laterality defects because of the way the cilia beat in the node being very similar to this circular motion. And when you look by electron microscopy, you see a number of findings. So you see a lot of normal looking cross sections. You see cross sections with a complete absence of the central pair. Sometimes you see double central pairs, and then sometimes you see one of these 9 outer microtubular doublets has gone into the middle and replaced the central pair, which is absent from the middle, and these tend to occur as you move up through the cilium. So this is sort of what I was saying about being in the base, the middle, or nearest the tip. So you tend to see, if you look longitudinally, the central pair is there, and then it disappears, and then another microtubular doublet comes in and replaces it as you're at the top. So when you're in the region of the microvilli, down here, things are quite normal. As you move up to about the middle, you see central pair absence. And then as you move more towards the top, you see this 8+1 appearance where an extra outer doublet comes in and moves into the center. So it's quite easy to miss if you're just looking at one plane of view within your sample, or if you're not looking at many cross sections within your sample. And this is another example here. So you've got a normal one here, but lots with an absence of the central pair where the clear white arrows are, and then microtubular doublets moving in towards the tip and making an 8+1 pattern. I'm going to go off on a bit of a tangent now and talk about electron tomography, and I'm going to do that because these defects are some of the first that were seen properly by electron tomography. So sometimes with those central complex defects, there is an awful lot of normal cross sections within them, and I used to think, well, it's a genetic defect, so why on earth are they not all abnormal if the gene's not there? And the answer to that really is that what we're seeing in the same as with an inner and outer Itodine arm defect, we're seeing lack of inner and outer arms due to problems with assembly genes. With central complex defects, we're seeing a problem with the central pair disappearing as a consequence of problems with the radial spoke head proteins. And interestingly, you can see the primary defect by electron microscopy. You just need a better technique and a better microscope. So I just wanted to introduce you a little bit to electron tomography, which is 3-dimensional electron microscopy. And my analogy here for traditional TEM versus tomography is, by traditional TEM, you're essentially seeing a shadow. So you're seeing a rabbit here. If you do tomography and you get a 3D image, then you actually get more detail about what's going on and the structure and the interaction of those structures. So you can see it's not a rabbit, it's a shadow or some hands. So how does electron tomography work? Essentially, you look at the sample in the same way as you would usually look at your sample, and then you tilt it. You can get anything to happen. Here we go. You tilt your sample in the microscope and you take a series of photographs from different angles. and then you can reconstruct all of those photographs into a tomogram, and then you can apply a series of averaging and mathematical techniques to try and enhance structures that way. And the first person to do this very well within human cilia and within PCD is the group of Daniela Nicastro in America, and they looked at patients with an RSPH1 defect. So that's people who end up with central complex defects by standard TEM. And as well as doing the tomography technique, they also use cryo-electron microscopy. And cryo-electron microscopy means that you're not limited by those chemical stains that we usually use, the heavy metals which reflect the electrons, and also by the agar and the aridite and things that we use in chemical fixation. So that improves the resolution in that way, and then the tomography also improves the resolution, and you're able to build a much better picture of what's going on in the cilia. And you can see in this patient with what would usually be a central complex defect by standard EM, using this technique, you see the same thing. So you see double central pairs, you see an absence of central pair, and you see this 8+1 arrangement with a microtubular doublet going into the middle. But if you look longitudinally, so if you're looking along that microtubular doublet, what you can see is that what is actually missing is the radial spoke head. So this is your microtubular doublet, these are your radial spokes, and there are 3 spokes for every 96 nanometers of the microtubular doublet. And you can see in people with RSPH1, or at least in this patient with RSPH1, these radial spoke heads are missing, and that can be seen by electron microscopy if you do cryo tomography. And this is just a model here, so you see all these secondary changes by standard electron microscopy, but this is the actual defect that is causing PCD. And I just wanted to highlight, so that paper now was quite a while ago. The electron microscopy techniques have moved on even more, and this was a paper that was published this week by Alan Brown in Harvard, but also lots of the BEAT-PCD collaborators, Jane Lucas, and this is just one of the figures, but really the resolution now using cryotomography, and this is bovine, so this is cow cilia, but it's still a mammal. You can see real detail of all of the structures that make up just a single microtubular doublet, all of the structures within the microtubular doublet, how they're interacting within different states of the ciliary beating. I would recommend it as a paper to anyone who's keen to know more, because really I think most of the proteins that make up the ciliary structure will probably be solved this way and will be able to be seen by electron microscopy, providing you have the time and the right equipment. So this takes us back to central pair defects. Some of the more subtle central pair defects are defects of the central pair projection. So this is a diagram of the central pair, and it has all these little projections that come off it, and there's an increasing number of genes now which when they're defective genes show absence of these projections. And by standard EM, you see a similar kind of thing to a central pair defect, only it's much more subtle. So you do see just one of the central pair present, you see this extra thing coming in. Sometimes the central pair look a little bit separated, and if you look very closely and you know what you're expecting to find, then you can see a sort of a boldness to the central microtubular doublet where there's not any structures on there. It's a tricky diagnosis. The high-speed video, as you can see, is not hugely abnormal. You don't see that same circling. You see a sort of semi-circling disorientation to the cilia as they're moving. And the genetics is also difficult because of HYDIN2, which is a copy gene. So when we first described HYDIN along with Heimat Omran's group in Germany, we actually applied the tomography technique, but not using cryotomography, just using our standard NHS machine. and we were able to make a 3D model essentially of the central pair, and showed that if once you've got better resolution, you can see the absence of that hydrogen structure. We still use that today in our clinical practice, and we also use an algorithm just to add lots and lots of central pairs on top of one another, and then perform averaging again to highlight the structures that are present and to highlight the structures that are absent. But really, the best or the most efficient way of diagnosing Hydian, I think, is highlighted by this paper, I think last year, from the OMRAN group, showing an absence of SPEF2 by antibody, and I think that has been the most helpful in our diagnostic practice beyond EM. But still, if we have a gene and we're unsure if it is a valid Hydian variant or not, then electron microscopy can help you to confirm if you can see that absence of the C2b, then it's likely to be PCD. So the next in the class 2 defects is mislocalization of basal bodies with few or no cilia. So we're not really talking about cilia here, we're talking about the lack of them. And with infection and inflammation, you do see a loss of cilia. But this is an example of a dead cell from a normal sample, and what you see even in dead cells, usually in a ciliated cell, is you see all of these basal bodies that are going to go and form the cilia, and they sit at the ciliary surface, and then the cilia form from those. So if you have lack of cilia because of an infective process, and we usually suggest that people don't do nasal brushings immediately following an acute upper respiratory tract infection, because you do lose the cilia. But in these aplasia cases, or in these reduced generation of multiple motile cilia, you see a complete absence of these basal bodies at the surface. So you can see here, there's no basal bodies at the surface. You see occasional ones within the cell. You can see the ciliary rootlet, the striated rootlet that I was talking about earlier in the normal samples, but you don't see cilia forming, and that can help in the diagnosis as to trying to tease out what's primary and what's secondary. So do we have a primary disorder where the person can't make cilia, or do we have a secondary disorder where there's lots and lots of infections? And I think that can be really hard to tease out sometimes, and therefore EM can be helpful within that context. And certainly we've got lots of patients that before these genes were identified, CCNO and Makaidas, which are two of the ciliogenesis genes, that had multiple brushings, because we just kept thinking that it was a bad brushing, and we didn't, the person had an infection, and actually the problem was lack of cilia. So we were never going to get an answer. So knowing that and being able to see that by EM is quite helpful. This is another example, which is the FOXJ1 gene. So this is a bit trickier, because actually you do get basal bodies that dock at the ciliary surface. So this is a control again, where you can see lots of nice basal bodies, but some of the basal bodies get stuck in the cell and don't make it to the surface, and that seems to be enough to cause disease. So it's certainly something to look out for in electron microscopy, with it, as well as looking at the cilia, to look within the cell and what's happening in the ciliated cells. Next is microtubular disorganization, and again that can be secondary to a number of different things, so it has to be taken within the context of, do you see it just in one cell, do you see it across multiple cells, do you see it across multiple brushings, or multiple sites, and does it make sense with the rest of the clinical history and the rest of the diagnostic tests. But this is just an example from two siblings who both saw the same changes, affecting about 31% of cross sections, counted 359, and 111 of them were disorganized. So that's the sort of disorganization I'm talking about, to be able to be helpful And by immunofluorescence, you have a lack of GAS8, which is the next indynine regulatory complex, and fits with these gene defects here. So again, if there's questions about these gene defects because you have a variant of unknown significance, or because there's no access to genetic testing, or you know, the next indynine regulatory complex, as shown by that work by Alan Brown and by others, is made of loads and loads of proteins, and there are going to be lots more genes in it. So it's useful to bear this in mind, that in conjunction with other findings, this could be PCD. And when we made the consensus guideline, interestingly, we weren't going to include this because you do see secondary disorganization quite a lot. But when we did our grid swap, it was one of the best things that people were good at identifying. So we decided that it could go in because as a group we were actually not too bad at it. And then the final one is absence of the outer and inner dyning arms, which I think we've all proved is recognizable, but only in a small number of cross sections, so in less than 50%, so 25 to 50% of the cross sections. And this is just a figure from the DNH9 gene paper, where there are different types of Itodine arm, one of which has DNH9 and the other which has DNH11. So if you're at the base of your cilia, you have DNH11 and DNH5, and at the top DNH9 and DNH5. And therefore, if you have a problem with this gene and with some of the other genes which form the docking complex, which only occur in the type 2 cilia, then you only see the changes in one region of the axoneme, and therefore it's important that you look at lots, but also important to think about the region you're in. And DNH9 defects, so this is in the distal cilium region, you can see there's an Itodine arm defect, and in the proximal cilium region the cilia look completely normal. So it doesn't make that criteria for an Itodine arm defect for the class 1 defects. And this is a number of patients that were in this paper, and you can see it's really quite variable. So this person, probably depending on where you are and where you count, only had about 20% of Itodine arm defects, whereas this person had about 90% of Itodine arm defects. So again, it's about thinking about where you are, what's happening with the cells, and thinking within the context of the whole sample and all of the diagnostic markers, rather than just looking at single cross sections from the axoneme. I thought I'd just include this because it is not in the diagnostic criteria, but it's another case of where reporting what you see by electron microscopy is useful, regardless if it's diagnostic or not, because once you put it together with the other tests, you can sometimes get a better idea of what's going on. So this is somebody with a defect in OFD1, and by light microscopy, you can see the cilia have these sort of bulbous tips sitting at the top of them, and when you look by electron microscopy, you can see that. So this is the cilium, and then this is a normal cilium next to it, and you see this huge bulb which is chocked full of microtubular doublets, and when you do immunofluorescence, contains lots of the intraflagellar transport proteins, and we've seen this now in a few people with OFD1, so can be useful. In the consensus statement, we didn't reach consensus on a number of other defects, and essentially that's why I was just highlighting, that it's still probably important to report those things, but they can't be currently called PCD due to lack of evidence in the literature, despite some people's experience. And those include things like the orientation of the cilia, and actually looking at the orientation can be useful because in a number of PCD defects, because all the cilia aren't beating together in the same way, you do see disorientation. It's not a specific sign. If you see outer arm or outer and inner arm defects in just a really, really small number, I think it's difficult to say that that's PCD. There's no evidence at the moment for length defects, and there's no evidence for isolated inner dyning arm defects in the same way if there's currently no inner dyning arm genes. And as I said before, there's been a lot of reporting of inner dyning arm defects, of things that were actually inner dyning arm and microtubular disorganization defects, so CCDC39 and CCDC40. There's also been quite a lot of reporting of inner dyning arm defects where there's an inner and outer dining arm defect, but the outer dining arm defect is partial, and therefore inner dining arm defect has been sort of written down or called because that's the major defect. And I think that's an important thing to keep in mind when you're looking at old literature and considering what's being said. So in this report, where we're covering everything we've ever seen, we also did as a consensus group, an idea of what we considered to be essentials to include, and also additional items which could enhance a report. So essential: whereabouts the sample came from, was it adequate for the diagnosis, how many cross sections had actually been assessed, what percentage of the cilia were abnormal, and did they have class 1 or class 2 defects, was the defect consistent across a number of cells, and then a one-sentence summary of key findings, which is sometimes something that as lab people we forget to do. Write down the opposite of not enough detail, lots and lots of detail. And then other things which I think I've probably covered in the talk, which are useful to enhance a report. So in summary, PCD diagnosis can be confirmed with EM with one of those 3 hallmark defects. Sensitivity is poor, and therefore EM can't be used to exclude PCD, but it can be used to support the diagnosis in conjunction with other diagnostic tests in a surprising number of cases, which might otherwise just be called normal ultrastructure because they're not hallmark defects. So can provide a useful basis for making a diagnosis in conjunction with the other tests. And the advanced EM techniques that allow improved resolution and structural defect detail are really coming on, and I think I would argue that there is no such thing as normal ultrastructure. Your microscope is just not good enough, because with the new microscopes you can see almost every protein that makes up a cilium, and if there's a defect within that or something that builds that, you will be able to see it. It's just in our clinical labs we don't have that capacity currently. And that's the end. I'll just leave you with these hallmark defects. Any questions? I'll unshare.
Thank you, Amelia, for your absolutely excellent talk. And it really didn't matter that most of your images were black and white, because it was still absolutely fascinating. So to kick off the Q&A session, we have a question from Regan. Do the doublets merge to form one, or is one longer than the other?
Oh, for the at the very tip. Yeah, no. Now, is it the A or is it the B? I can't remember. but no, you just one stops and the other carries on, and they don't necessarily all do it exactly at the same time as well. So you do see some cross sections that have got a few doublets and a few singlets in that sort of interim area.
Thank you.
There's another question about the tomography. So Stefan is asking, how often do you use tomography for regular diagnostics, and what about costs and time and effort?
Yeah, so compared to doing regular EM, the tomography is really involved. It takes a couple of hours just to collect all of the images, because you have to collect whatever 140 images to make the 3D tomogram, and then the computer program in order to realign those isn't as automated as you'd hope. So there's a good day's work for one sample, if not more, in order to do the tomography. So we certainly don't do it in all cases. We have what we call our difficult case list, where we find it difficult to say, does the person have PCD, does the person not have PCD, and that's usually based on some high-speed video microscopy findings that haven't gone away in culture, or a really strong clinical history that means the person keeps coming back, but negative genetics and negative on EM. And it's those cases that we will do the tomography. Or, for example, for hide-in, if we get back a variant of unknown significance from the genetics lab, then we would do the tomography in order to be able to confirm or exclude that. I didn't show the data for DNH11 because it's published, and I feel like people might be sick of hearing me go on about it. But we also do it for occasional DNH11 cases, because you can see there's a absence of structure at the base of the cilium in DNH11. So again, patients where we might find one pathogenic variant and no second variant in DNH11 is useful, and the ultrastructure is normal, and we think from the high-speed video that it's a DNH11 type pattern. It's been shown that the antibodies for DNH11 don't always capture it, so you can have DNH11 present on antibodies sometimes. So those kinds of cases, so it's rare cases, and we're also finding that some of the things we used to do by tomography, we have an averaging program where essentially you overlay lots and lots of cross sections over one another. So without doing the 3D items, and hide-in particularly, we use that quite a lot. So you just overlay and then take an average of lots of cross sections all put together. And we've published that as well. It's called PCD Aid. It's Tom Burgoyne wrote that, and that's quite useful for a quicker version of the tomography.
Thank you. We have 3 questions from one person here, so we'll take them one at a time. Thanks for the great talk, very useful to me and my colleagues. Are there a lot of EM images showing typical abnormalities that are available somewhere as self-training materials for beginners?
Yes. So I can answer that. So yeah, we have some on the BEAT-PCD website under resources, and I might be able to post the link to that in a minute actually. So they're all in folders with what the gene defect is, and they're not just from my lab, they're from a number of different labs that took part in the audit for the consensus statement with permission. So I think they give quite a good idea of what the different defects look like, with different processing techniques taken on different microscopes, and they're available to use.
This person also says that they remember that some European researchers are trying to establish a machine learning system for interpretation of EM abnormalities of cilia. Is it used practically?
Yes. Is Andrea on the call? Is Andrea here?
I've seen her name. Oh, I think she might have gone.
She's gone. Okay, so yeah, we at the moment are trying to develop in the Brompton an AI system, and it's being led by Andrea Pinto, who was here earlier. And I didn't talk much about it because I think in next year's programme there's potentially a whole talk about it. But yeah, so we've been working with a company who've now been taken over by Intel, doing AI on EM images, and it's a user in the loop system. So essentially what the AI does is it's able to screen out lots and lots of normal cross sections. So we take the images and then input them into the system, and then it highlights the ones that are abnormal. Andrea is beginning to get to a point that she can train it to recognize hide-in, which is neat, and it puts a little box around where it thinks the defect is. And we've tested it now against a number of experts. So we've tested it against 5 experts in the UK, and it's as good as we are, and more certain about its conclusions, I think, is one thing that's interesting. So that AI, so where sometimes we sit on the fence and say, oh, well, it might be abnormal. The AI makes a decision either way. So we are developing that. At the moment, we're running it along with our MDT meeting. So the AI comes to the MDT meeting and gives its opinion as well, and we compare it to the MDT opinion. So that's happening. But Andrea is probably better placed to speak about it as she's leading it.
And the third question is the sharpness of EM images affected by how the specimen is collected? That's
a good question. I don't think it's the major factor. To be honest, I don't have huge experience of working with curettes. Our brushings and our ALI are fairly similar. in terms of cultures. I guess probably the processing and the staining are probably bigger factors, and maybe if you've got a thicker biopsy or something, then you could potentially the protocol might end up being different, and therefore you might get a different sharpness. That would be my opinion.
Okay, thank you. Nuria is asking, first she's thanking you for the talk, and she's wondering, is it recommended worth to evaluate the transition zone with the TEM? So for example, with RPGR defects, and then maybe analyzing the orientation, as you mentioned at the end of your talk.
So I've not to date seen a transition zone defect that's recognizable, but there's no harm in having a look, is there? Because the more you start to understand what's normal, the more you start to recognize things which are abnormal. But for the RPG, and for the RPGR defects, I assume you're referring to Vito's paper. I don't know if there's anyone from Southampton here who wants to comment on that any further. No. So my answer is I can't recognize an RPGR defect from the transition zone, but I only have a couple of patients that I'm aware of. Oh, Claire can comment.
Can you hear me?
Yeah.
Oh, sorry. I'm sat in a car with my daughter asleep in the back seat. So yeah, I just wanted to add that RPGR, I think, is quite recognisable, possibly more from the high-speed video point of view, because you get different lengths of cilia along the ciliated edge. And then I think is quite a strong feature going into the diagnostic process, and it really supports any findings you might then see on the EM. So that's my only comment behind that. That's something that we did talk about a lot as part of that study.
Thank you.
So the next question is from Elif. Dear Amelia, thanks for a great talk. We still have problems with sample processing. One big problem is the thickness of the sections, and also the other one is losing volume of the sample during steps. Do you have any recommendation about this?
Yes. What I was taught to do, it doesn't necessarily mean it's the right thing to do, but what I was taught to do was to to prevent losing the sample is fairly early on in the processing, to spin it down, to centrifuge it, and then to centrifuge it in molten agar. And then you can treat it as a piece of tissue. So I centrifuge it down in molten agar, and it's a technique borrowed from looking at bacteria and things like that. So instead of having lots and lots of single cells and strips that you're trying to fish out and not lose, you can centrifuge it in molten agar, give it a little stir up, and then set it in the fridge for about 5 minutes, and then you can treat it as a piece of tissue because it's all embedded with agar, and then you can do your dehydration steps in that piece of agar without losing things every time you change. So I do the fixation, a spin, and osmium and a spin, and then do the agar step, and then do the rest of the processing with it embedded in agar. And I think that prevents losing some of the sample. Question about the thickness of sections. I tend to cut relatively thick compared to what you might do for other EM, and I do that because we know that the structure repeats every 96 nanometers of the dining arms of the radial spokes. So if your section is too thin, then you're always going to miss something because you haven't got the repeat of that structure. So I tend to cut sort of pale gold-ish sections on the reflection rather than a silver section that you might do if you had a kidney biopsy or something like that.
That's really interesting. So we have another question from Monica Burkhead. How long would you spend on a difficult specimen, so hardly any cilia cells, hardly any ciliated cells present, when resampling could only be done several months later at the next clinic visit? So how much time and effort would you spend on one sample?
Honestly, there's some samples that don't get past the toll blue. So we'll look at the toll blue and have nothing, and keep sectioning through on the sort of the toll blue, for anyone who doesn't know, is the sort of light microscopy section you take to decide where to take your smaller electron microscopy section. So you take some sections off and try and find the best area in order to then take a much smaller section, because the electron microscope will only take a small section. So sometimes I don't even go to electron microscopy. If I always make a few blocks, and I always cut all of the blocks that have been made, and always cut a couple of toll blues from each block. But if there's nothing there, then I don't persist. We also have a rule when we do our light microscopy that if we can't get 10 strips from, I think it's 4 slides, that are ciliated, then we don't even fix it for electron microscopy, because it's a waste of everybody's time, because it is literally like looking for a needle in a haystack. If you can't see it with a big light microscope, then the chances of finding it with an electron microscope are really slim. And we do a lot of repeat brushings, and I would never do a repeat brushing if I could help it, but quite often it's not possible to get a result by electron microscopy. And if you do get a result, you know, if you cut section after section after section to get your 50 cilia, it's usually really unhealthy, and you usually don't get a certain diagnosis, because there's been so few cilia there, they carry so many secondary defects that it's impossible to say PCD or not. So not very long, I think, is the answer.
Can't see any more questions in the chat. Here's one. I sometimes see clusters of tip-looking cross-sections on the height of microvilli in otherwise completely well-looking samples. Is an interpretation of those as growing cilia a possibility?
Yeah, I think that's a good possibility. Growing cilia, or sort of bent over cilia sometimes. So sometimes once you've done your processing and things are in different orientation, sometimes cilia that have come from a different cell, because you're looking at a very, very thin section. So sometimes you can have another cell that's underneath that you can't see by the electron microscope, but the tips are coming up through. But yeah, growing cilia is also definitely an explanation, I think.
So if there are no more questions, I think we'll wrap up here. Thank you, Amelia, for an absolutely excellent talk. We really